I'll start by sharing my screen, and I want to be sure that you all can see it.
Perfect, Val.
Is it in full screen?
Not yet. Yeah.
Okay, great.
Val, one second. Some people still have their microphones on. Please mute your microphone, other people.
Hi, Greg. So yes, thank you to Greg as well, as one of the organizers of the meeting, for having me speak here. I'm glad to hear that you could join us. Christina was mentioning earlier that you felt a little ill this morning, so glad you're here, Greg.
Well, I'm going to stay here without video. You don't want to see me.
Okay, so let me just move this bar of, I don't know how to do this here. Just give me 1 second. Okay, great. All right, so the title that I gave to the talk today is Democratizing Multiplex Imaging Using Formalin-Fixed Paraffin-Embedded Samples and Open Source Tools. This is essentially what I went to do at the NIH earlier this year. All of us know that we do have access to many samples that are paraffin-embedded in different pathology labs at universities, in private hospitals, public hospitals. But all of us also know that it's really a challenge to be able to do multiplex imaging in paraffin-embedded sections. So when I heard of this possibility, I immediately wanted to learn it, to bring it to our lab, but also to share the technology with the Brazilian community and anyone that would be interested in it. So, so that's what I'm going to show you today. But before then, I just wanted to mention to you that, as Christina said in her presentation, our lab has been focusing on studying the immune mechanisms associated to the establishment of protective and pathogenic immune responses in parasitic diseases, particularly leishmaniasis and Chagas disease. For those of you who are not familiar with these two parasitic diseases, leishmaniasis is caused by the infection with different species of leishmania. In our case, we study particularly the tegumentary forms of leishmaniasis that are caused by Leishmania brasiliensis in an endemic area in Bahia. And here, I'm not going to go into details of the life cycle of the parasite, but I just wanted to point out that leishmaniasis can be transmitted by the sandfly during its blood meal in human hosts. And within the host, the parasite can go into different cycles of multiplication, can spread into the tissues, especially in the case of tegumentary leishmaniasis, the skin or mucosal sites in the body. And then these parasites can also infected cells can also reach the bloodstream and be picked up by mosquitoes, by sandflies that would then transmit it back again. Right here on the left side of the figure, I show you the manifestations of cutaneous leishmaniasis, which is typically a skin lesion, as you see here, and the mucosal form, which can affect different mucosal tissues. They're both, these are pretty mild forms of representation that I chose to show you today, but they can be very mutilating and very traumatizing to the people that bear them. The other disease that we've been studying for many years is Chagas disease, that is caused by a cousin of Leishmania, which is another parasite from the same family, which is Trypanosoma cruzi. Trypanosoma cruzi is also transmitted during the blood meal by the feces and urine of contaminated feces and urine of the kissing bug or the barbeiro. And this parasite also goes through a cycle inside of the host, where it typically chooses particular cell types to live in. It is a pretty promiscuous parasite, but in the case of most species of most types of trypanosoma, they choose to live in muscle cells, particularly heart muscle cells. And this causes the most severe manifestation of Chagas disease. Of course, there are others. The parasite can also go into the digestive tract and cause other manifestations, but the most severe form really is the cardiac clinical form that is represented here by the enlargement of the heart that can lead to sudden death and heart failure So it's a very severe disease. In the lab, we have been studying what causes the manifestations in leishmaniasis, both cutaneous and mucosal, but also in Chagas disease. And these two diseases, the interesting thing about them is that despite having the severe manifestations of the disease, they also manifest as less severe clinical forms. So the idea is to try to understand why some people develop less severe and other people develop very severe clinical forms of both Chagas and leishmaniasis. Typically, we study this response in the peripheral blood of the patients. We have done many studies in situ in the case of leishmaniasis with students from the cell biology graduate program that both Christina and Greg are part of, defining the mechanisms of tissue destruction in different forms of leishmaniasis because these lesions are accessible, right? So it's easier to study them. In the case of Chagas disease, of course, it's a little bit more challenging because you have to have access to hearts of people that either underwent transplantation or died of Chagas disease. So typically we study the blood of these individuals to try to get a picture of what's going on with the patients during the different manifestations of the disease. Our focus in both Chagas disease and leishmaniasis has been to study the mechanisms associated to the immune regulation in these patients. So if you look at this picture here, you see a cell in the center that is an antigen presenting cell. In both the cases of Leishmania and Trypanosoma cruzi, both these parasites are able to parasitize the cell. And it's very important to remember that at the beginning of the infection, during the acute infection with Chagas, with Trypanosoma cruzi and Leishmania, these cells have to be activated to control the parasite. And this control is something that really happens very efficiently in the human host. So although you have acute phases that are characterized by very high parasitemia, as the disease progresses a little bit, these cells are activated and can control these parasites to very low levels in both Chagas and leishmaniasis. And this activation of these cells rely on the production of what we call inflammatory cytokines. And two examples are here, TNF-alpha and gamma interferon, that are produced by many different cell types in the host, immune cells. And these cytokines are able to activate these parasite-bearing cells to kill the parasite. So this is an important moment. This initial inflammation, this initial activation is very important for controlling diseases. And I'm giving you the example of Chagas and leishmaniasis, but this is also true for many other parasitic infections and also viral infections as well. However, if this inflammatory environment that is so important at the beginning is not controlled, it can cause tissue destruction and it can cause serious pathology. So an exacerbated immune response, an uncontrolled immune response can cause tissue pathology, which can be very detrimental to the host. So that is where the other side of the immune response, represented here in green, that I call the regulatory environment, takes place, and it's very important. So you have here the representation of especially IL-10, that is an anti-inflammatory cytokine, that the production of this cytokine can then control this inflammatory response. I showed to you here the yin and yang, showing the importance of having an inflammation on one side and the control of the inflammation on the other side. Typically, what we see in the mild forms of these parasitic diseases is this balance between the control of parasite and the lack of tissue destruction. If by any reason this balance is broken and it's shifted toward a more inflammatory profile, this will be very important to cause tissue destruction, and therefore that's what we would like to avoid. We wanted to keep people with a balanced response in a way that they don't have tissue destruction and severe forms of these diseases. So the main question that we've been asking in the lab for many years is how to apply the knowledge generated by the study of these immunoregulatory networks to drive new therapeutic interventions for parasitic diseases. So we believe that by understanding the cytokine balance during the different forms of the disease, and also by knowing the cellular sources of these different cytokines, by identifying the sources, we can provide means of controlling and exacerbated response and control pathology. So that's what, in general, we've been trying to do in the lab. And as I mentioned to you, we've been studying the peripheral blood. And I think this figure here shows to us the connection that exists between the peripheral blood and the site of infection and the place where you do see the inflammation and tissue destruction in these diseases. So this example here is given by the infection with Leishmania, but it is also true for the infection with trypanosome and, like I said, many other parasites as well as viral infections, right? So we've been studying the peripheral blood response, but we also very much wanted to understand and still want to understand what happens at the site of lesion, what happens in situ, where the tissue destruction is taking place. And the reason why it's important to understand that is because by doing so, we can then try to monitor this response, we can try to control this response and avoid the tissue destruction. So essentially what happens is that when you have an infection, these parasites will then enter several different cell types, amongst them the antigen-presenting cells that I mentioned to you. These cells will migrate to secondary lymphoid organs. They will become activated in these secondary lymphoid organs. And also there, they will activate antigen-specific cells, parasite-specific cells, T cells and B cells. These cells will then gain the bloodstream. And because of the changes that happen in their surface upon activation in the secondary lymphoid organs, these cells can then be recruited to the site of inflammation, as the endothelia in these regions will be modified and will be prone to interact with activated cells by these different parasites. These cells will then transmigrate to the tissue, and this is what will lead to the inflammation. This will happen many, many, many times, and this cycle of migration and recruitment is exactly what will cause the inflammation. As you can see here in leishmaniasis, this inflammation will manifest through the appearance of a skin lesion, and will also appear in mucosal lesions. And in the case of Chagas disease, what happens with the heart is that this inflammation will trigger the establishment of fibrosis. This excessive fibrosis will lead to a very severe burden in the heart muscle, and this will lead eventually to dilation and potentially the death of these patients. So to understand what happens in the place where the disease actually manifests itself, the way to do it is to take lesions from the skin, from the mucosal site in the case of leishmaniasis, or parts of the heart of the patients with Chagas disease, and try to stain and characterize the cells that are mediating the tissue destruction, that compose the inflammatory infiltrate. And ideally, we would do it employing multi-parameter analysis that will allow for us to identify many different components of the response at once. However, as nice as this may sound and as interesting this may sound, this poses many, many, many challenges to us, especially when you think that it's difficult to get these samples, it's difficult to get these organs, and the material that most of us have access to is paraffin-embedded tissue. When you think of fluorescent, using fluorescence probes in paraffin-embedded tissue, this really poses many, many challenges and can be quite difficult. I can tell you that wait a second here. What have I done here? Can you still see it? Okay, that's back now. Apologies for that.
Perfect.
So, when you think of using fluorescence to identify different cells and components present in the site of tissue destruction, and if the material that you have is paraffin-embedded tissue, this really poses many, many challenges to us. And that's what I wanted to discuss with you today. Thinking about that, and I can tell you that since many years ago, we have tried extensively doing fluorescence, multi-parameter fluorescence in tissues from paraffin-embedded the repositories, and it really has been a major tragedy. We were incapable of doing that. Then I came across a couple of papers, actually one paper at first that came out in Nature Protocols that was performed by a group at the NIH, headed by Ron Germain, and the first author, the person who really established the methodology, is Andrea Radke. So she was the first author of this paper. We had seen other papers that used similar methodologies, but not quite as straightforward as this one. So we came across these two methodology papers that are shown here to you. One came out in a cover of Nature Methods and one in a cover of Nature Protocols recently. And these are essentially fluorescence images. This is obviously not true. She made sort of like a church window or something like that. But these were images that she acquired using a methodology that allows for you to do multi-parameter analysis in tissues in different human and experimental derived tissues. So once we came across that methodology, we were very interested in seeing if we could apply it for our needs here in Brazil. So what is this methodology? So IBEX is what Andrea calls it, stands for Iterative Immunolabeling and Chemical Bleaching Method for High Content Imaging of Diverse Tissues. So essentially what she does is she established two different types of methodology, one that she calls manual IBEX and one that she calls automated IBEX, that allows for you to take one section of tissue, label it. This is a very summarized scheme of this method. So essentially what it allows for you to do is to take a section of your tissue, label it with different antibodies, as you can see here, each antibody is stained with a different fluorochrome. Then after labeling, you image it using a confocal microscope. Then you bleach it, and I'll give you more details in a minute about this, and then you restain it. So essentially what Andrea did was she established many different cycles using the same slice of tissue, either manually or using an automated system. This automated system, essentially the difference of it from the manual is that right here, from the moment you put your tissue there to remove the paraffin, to go through all the steps, blockings, labeling and all that, it's done automatically by a machine. Obviously, this is a very nice strategy, but for us, it wasn't as interesting because the machine that does it for you automatically costs almost $1 million, and that would be prohibitive for us at the moment here in our lab. So we were very interested in doing the manual and studying and establishing the manual strategy for the IBEX. So what to do is you image your tissue, and if you have many different cycles, for example, in this example here, you have 4 different cycles of staining. What you do is that you later use computer programs and algorithms to align the samples that you took from the very same slice, and then you end up with a number of different markers in the same tissue in the same place. So we were really very interested in that. However, in the two papers of Andrea and the subsequent ones that she published, what she uses mostly, even though she standardized the manual and the automated, typically she uses more of the automated, and she uses samples that are frozen. Although she does have some paraffin embedded, but most of them, if you look at the paper, will be frozen samples. So we were still very interested in that because she did have some that were paraffin embedded and used in the manual system. So we wanted to try to learn that and bring it to Brazil. This is just to show you some of the images that she obtained in one of her papers. This is a human lymph node. She stained here a total of 38 parameters. So as you can see here, she has here the accumulation of several of these parameters. And here she has broken them into different combinations, depending on what the question that she was asking was. But as you can see, you can see a huge number of different molecules, in this case, 38 different molecules in the same section from the same tissue with different colors. Essentially, obviously, what you do is you tell the computer, once you acquire the images, what color you want to call each fluorochrome, and then you go stacking them and establishing this association in the same region of the tissue. So you can clearly see very well-defined regions here. So in this human lymph node, you can see here regions that are rich on B cells, for example. Again, a co-staining with another B cell marker. So it's very, here outside of the B cell region, you can see a T cell rich region. Similarly here, you can see a few T cells spread through the lymph node. So you can clearly have a very good idea of the distribution of different cell types in this lymph node. And obviously, you can also use markers that are functionally expressed by different cell types. For example, FOXP3, as you know, is expressed by regulatory T cells. So you can see T cells that express CD3 as well as FOXP3, for example. You can also use markers for other structures that you find in the lymph node, not only the cell populations, but you can stain for blood vessels and also for lymphatic vessels in this tissue. So it's a very powerful methodology that allows for you to identify cell populations, functional characteristics of these populations, how the cells interact with one another, but also extracellular matrix and vascular characteristics of the different tissues. Here's another example of the human spleen that she was able to do 25 parameters using the manual IBEX, but in frozen samples, similarly to the previous one. So you can clearly see the staining again for the different populations. Here she has a number of extracellular matrix markers that allows for you to see the organization of the tissue really well. Here you have macrophage markers, again, extracellular matrix markers, T cell markers, activation molecules, functional molecules, and so on and so forth. So essentially, you can really have a very good idea of what's happening in physiologic conditions like she did, but you can also have a good idea of what happens with these tissues upon an infection or a disease, for example. Here again, another example. This is a beautiful image of a human liver, again, done by manual IBEX, not using the expensive machine, and frozen samples. And you have a marker here for lymphatics as well. You have a marker for activation of different cells, and essentially you can identify many different components of the extracellular matrix in this tissue as well. So you can study it not only by the composition of the cells, but also structurally. The technique allows for you to really see very well what's going on. So here, again, just to remind you, this is really excellent. I had the opportunity to see this machine working in Andrea's lab when I was at the NIH in January. So essentially, it's an awesome system, but it requires this machine that will do everything for you. So essentially, you put your antibodies, you put your buffers, your blocking buffer, your washing buffer, and everything. And this comes with a chamber where you can put your tissue, and everything will happen here. Once you introduce it in the machine, everything will happen in here. All you have to do later is read it in the microscope, which can also be transferred here. And as you go cycling it, after you finish your first cycle, after you read it, once you have to do your bleaching and your secondary stainings and all that, you can do all these in this machine. Fantastic. You can take only 12 hours to do up to 28 markers in 4 different cycles. But like I said, for us, it's not realistic to think of this machine at the moment, unfortunately. This figure here I just put to show you a brief comparison between the images that she gets from the automated IBEX which is the one using the machine from the very beginning, with the manual IBEX. And I'm not calling attention to particular molecules here because they are in different colors in the two comparisons. But as you can see, the intensity of the staining that you see, the organization that you see of the different structures in the automated system is very much similar to what you see in the manual IBEX. So in other words, the automated system is great because you can put it, you can leave it overnight, you can go home, you can sleep, and by the next morning when you get there, everything is going to be ready for you. But you can also do it manually with a very, very good quality of staining that is pretty much comparable to what you get with the automated system. This is the little machine here. It's amazing to think of the price of these tiny little things, right? But you have it here, and then and the price of that is not counting on a microscope, obviously. So this is the system that uses the ARIA system that does the automatic staining for you. But like I said, it's pretty much comparable to what to do with the manual thing. One aspect that is critical for you to achieve the many different cycles. So really the nice thing about this methodology is not only that you can stain up to 6 or 7 molecules in the first cycle, and what's going to determine the number of molecules that you can stain per cycle essentially is going to be the capacity of the confocal microscope that you have. If you have a confocal microscope that can read 3 parameters, you can stain for 3 parameters, bleach it, do a second cycle in 3 parameters, and do it as many times as you want. If you have a confocal that allows for you to read 6 or 7 or 8 parameters, you can do your first cycle with as many parameters as your confocal reads. You can then bleach your fluorochromes and come with the second cycle on top of that. So that is what is going to determine how many molecules you're going to stain and how many cycles you're going to have to do depending on what molecules you want to do. But two very important things here to show you in this slide. The first one is that it is critical that you bleach your cycle once you read it perfectly. So essentially what the IBEX does is it allows for you to stain the molecules that you want in your first cycle. You collect that image, then you come with a chemical that is lithium borohydrate. And it's a very potent reducing agent that will bleach your fluorochromes completely, right? So once you bleach it, you can come with your second round of antibodies to stain your tissue, and then you will acquire it again, and then you bleach it again, and you come with your third cycle, and so on and so forth. So this bleaching really is absolutely critical, and I'll show you some images of the bleaching that we've had when we were at the NIH. But this just shows you here that if you bleach it properly, you can see the staining in cycle 1, for example, for PD1 with this distribution. In cycle 2, you come with a different molecule in different cells, and you see a different distribution. And then in the third cycle, you come with a different molecule, and it will allow for you to see a different distribution. So this is one good tip that we use when we are doing IBEX. It is good that you use in each cycle, if you're using the same fluorochrome, that you use molecules that are not concurrent in the same cell. They can be seen in different cells, because that will help you determine, even though you're going to look in a microscope and see if you bleached it well, but that will be you'll be sure that you're not going to have overlapping in the same cell type. So this is one important thing. Bleaching is absolutely critical for you having a good staining in each cycle that you do. The other thing that is very critical is what is shown here in these two images in the middle here, the JoJo 1 and the CD4AF594. This is what Andrea and people that work with this methodology call the fiducial markers. These markers essentially are markers that are not bleachable. So you use things that will not be bleached by the lithium borohydrate, and you need something that stays identical in all the different cycles that you use, because you're going to use these different molecules, these different markers as your guides to then when you go overlap the different cycles, you use them to guide where you were in the tissue to be sure that you are overlapping the different cycles in the very same place. So this is absolutely critical. It's very important that you maintain these fiducial markers here. This graph here on the left shows you the time that takes to bleach different fluorochromes using lithium borohydrate and sometimes in combination with light. As you see here, if you use the BV markers, you have to use light together with the lithium borohydrate to be able to bleach it really well. So you can do that in about 10 minutes. Other fluorochromes are much more susceptible to the lithium borohydrate, so you don't have to worry about it. Others are very resistant to it. And in fact, for example, AF594 is one that is not bleachable at all using this chemical. So this is a good fluorochrome for you to use with one of your fiducial markers. So that will stay through your cycles, as you see shown here, because this fluorochrome is not susceptible to the bleaching by the lithium borohydrate. So this is a good way of choosing it. As you see here in this graph on the right, most of the fluorochromes will disappear after at least 5 minutes of incubation with lithium borohydrate, but some of them will not. And those are the ones that you want to use for your fiducial markers. In other words, the markers that will stay there in all the cycles in the very same place that will allow for you to then align them properly, okay? So this bleaching is very critical. And another important information, especially for us who work with parasites and have sometimes the opportunity of using parasites that are, or other proteins that are directly labeled with green fluorescent protein, red fluorescent protein, and so on and so forth, is the fact that these proteins are not bleachable using lithium borohydrate. So this is, you either use them for your fiducial marker, or if you are to use them, use always in your last cycle when you don't care about bleaching anymore, because they do not bleach really well. Another important thing that Andrea showed in this very potent methodology is a comparison between the analysis of expression of many different markers in B cells, in T cells, in myeloid cells, and in structural molecules, comparing what she called the serial IBEX, which is the one that uses just one slice and different cycles, versus the one that you do in serial slices. So you would do each one of these molecules in one slice, serial slice from the same tissue. And as you can see, this expression is quite comparable for most molecules, showing that you can really see an excellent expression of the different molecules using both methodologies, which means that obviously using the one slice of tissue will give you an easier and more reliable way of doing it. And of course, will also save material. Sometimes you can only get access to one slice of the tissue. Perhaps this will be used for other diagnostics and things like that, and you don't have access to many different slices. So using one slice also provides you with the opportunity of staining many different markers without using up a lot of your material. Now, what do you do once you have all your images stained and aligned and read? What you have to do is you have to properly align the images that you obtain from each different cycle. So once you have your images collected from your confocal microscope that are stored in your storing system in your confocal microscope, and you need to align them, what is critical here is to remember that you will rely on what I mentioned to you as your fiducial marker. So for example, you have the expression of your fiducial markers that you know would be in the same place, and then you will use that to be able to align. So you're going to lose the blurriness of the many different markers that you have once you put the fiducials right on top of one another, and that will resolve the staining for you. So that is a critical moment. This alignment is very, very important, and it can be done either with commercial softwares, but it can also be done with open source tools that in the case of our use here is a lot more feasible, because the software that uses this is quite expensive. If you want to use the commercial one, is quite expensive. And again, that can be a problem for us. So using the open source tools and algorithms really allow for you to align it properly, to be sure that you're stacking your cells correctly. Another important thing that I can tell you from our experience there is when you're reading your sample in your confocal microscope, you should look for things like this. You see this image here in the letter B. I don't know if you see my pointer. Can you see my pointer? Can you see my computer pointer here?
Yes, yes.
Okay, great. So when you're collecting your sample, you look for things like this that are weird images that you can compare between the two different cycles. And that will help you at least guide at the beginning where you're going to align your samples. And of course, when you're going to acquire, if you're not acquiring the entire tissue, you can choose the regions that have the funny structures or the marker structures that will allow for you to then align them properly later. Just as a reminder, it's often, for example, when I was reading the Chagas heart samples, we read about 5 fields per sample, and that took about 40 to 50 minutes per sample to read. So it takes a long time, because remember, you have as many channels as you want to use. So if you have 7 channels, you read each slice for each of the channels that you're acquiring. So it's a quite time-consuming thing. So you want to be sure that when you analyze, when you acquire the subsequent cycles, you will acquire in the best possible place to help you align it later on with the software. So the software really is very good. The algorithm was developed by Ziv, who is a person that also works at the NIH with Andrea, and it's very powerful. So even if you're not as careful as we were, because we were guided by her, who had a lot of experience with it, it can still align your tissue for you. The danger with that is that if you don't be sure and acquire in approximately the same region for everything, is that perhaps sometimes you can align correctly with the software, but you're going to lose a lot of tissue that is non-aligned if you acquire in different in regions that are very, very different, right? So it's important to be aware of these things. This is all really well explained in Andrea's paper, and we are also writing our own paper to describe all this methodology that we've established there. All right, so once you have your many markers, look at this here, she has many different markers in the same section of the tissue that she stained, acquired, aligned. What do you do with it? How do you analyze this? So it's a number of different molecules that you want to see what cells are expression, the co-expression of these different molecules, the association of the different cell types, and so on and so forth. So one of the methodologies that you can use to analyze these types of data is this one that you analyze, people have been using now for a long time for analyzing complex multi-parameter obtained data, which is the T-distributed stochastic neighbor embedding. We use it a lot for flow cytometry currently. It's a very powerful type of analysis because you can just feed your data into the program and it will analyze it for you without any bias. So it will distribute the populations for you based on the expression of each one of your markers in a heat map type thing, showing you where such molecule is more or less expressed and where there is co-expression of different molecules. You can look at that, but it will also give you an idea of the expression of as many molecules as you have in your staining. And this can obviously all be translated into numbers, into pixels, into intensity, into frequency, and you can analyze it statistically. So it's a very powerful way of doing this. You can also use all the programs that we typically use for quantifying cells and for quantifying intensity. You don't need to do it in an unbiased manner. If you know what you're looking for, you can also do that. But this also gives you and points to you things that perhaps we're not looking for, perhaps we're not thinking about. So this will give you excellent idea of what you're doing. Okay, so this is all great. This is all excellent what Andrea did with the frozen samples and the manual IBEX. So for us, working with human paraffin embedded formalin fixed samples, the issue is that you have to deal with standard preparations that are used in pathology labs. Typically, this way of fixing and embedding leads to very high background. As I mentioned to you, we've tried extensively and always had extremely high background, very difficult to clear. And of course, if you're working in resource-constrained areas and countries, the cost of automated systems just make it completely prohibitive. Maintenance and cost of equipments, delay of buying complex machines and maintenance for them, reagent availability, variety of types of confocal microscopes that you have access to. So these are all challenges that we have to deal with when we are thinking about how we are going to implement it here. But fortunately, I think now we are able to overcome some of these challenges. And when I went to the NIH through the CNPq Estágio Sênior Fellowship for January and February of this year, which was actually, my stage was cut short for 15 days because I got COVID in early January. So I was fine, but the NIH wouldn't let me step into the lab, while I was positive for COVID, and then you have to wait a little bit. But our goal was really to concentrate on developing a manual IBEX used for human FFPE samples. So what we did is we took some samples with us from Brazil that were from pathology labs. In the case of Chagas, through a collaboration with Professor Geraldo Brasileiro at the medical school at UFMG. And we also had some cancer samples in collaboration with the Hospital Mário Pena in Belo Horizonte, and also some leishmania samples from the University Hospital in Bahia. So we took the samples and we went to Andrea's lab to try to establish this methodology there. So this is essentially what you do, what we did when we were there. First thing that we did is we tried to establish the method with tonsils, human tonsils that we had that were prepared in the very same way, just so we wouldn't use up our patient samples. So these were materials that we had from people that underwent tonsillectomy surgeries. So we used those tonsils to establish the methodology to test our antibodies and all that. We test many different new antibodies and antibody combinations to use. We also designed the different sequences and the different cycles to try to minimize use antibodies that are labeled with fluorochromes that would minimize spectral overlap, and at the same time optimize useful information, and I'll talk a little bit about that soon. And then we ran the experiments with the different cell types. So to do that, we were hosted by Giorgio Trincheri at the National Cancer Institute, who is a long-term collaborator of ours, Dr. David Sachs, who many of you know, it's an expert in leishmaniasis, so he also hosted us in his lab. Andrea, who is the person that established the methodology, and a very key person who was Sundar Ganeshan, who is the Leica SP8 operator at the NIAID Building 4, where we were, that was truly instrumental for helping us read all the samples that sometimes took 12 to 13 hours to read. So he truly allowed for us to be there in the facility reading the samples. So we used cervical cancer samples, as I mentioned, cutaneous leishmaniasis, and Chagas disease cardiomyopathy samples, all using fluorescent labeled different monoclonal antibodies and the different cycles for IBEX. So the first thing that you have to do, if you're dealing with paraffin-embedded tissue, is to be sure that the tissue that you use have a maintained structure, right? So first thing that we did is we took all of our samples and we just basically cut a slice out of them and did a hematoxylin using plain staining, just to take a look at the tissue and see if they were usable, if they were okay, if their structure was good. And as you can see, some of them we had, for example, 8 Chagas heart, but we only were able to use 6 of them because 2 really had a quite hurt structure for whatever reason, you know, many reasons for that. Some of the samples are quite old, have been stored for a long time. So, you know, not always you can have 100% of your samples useful to doing the IBEX. So after we selected the different samples, we did what IBEX does. We did the cycling, the first cycle, we bleached, we collected, we first cycle, collected, bleached, second cycle, and then we did the analysis. So this is a little bit more detail of a protocol. So essentially what you do is you cut a 0.5 micrometer slice of your tissue, you let it dry overnight at 65 degrees, then you remove paraffin and do your antigen recovery. For the antigen recovery, we use this buffer that is commercially available. That is a pH 6 antigen recovery buffer. That so you put your samples there and you boil it for 30 minutes at 90 degrees and it recovers antigens beautifully. Then after that, you do your blocking step using BSA and FC block for the monoclonal antibodies. Then you start your stainings with your cycle 1 with the markers that you chose and want, always remembering to have one fiducial marker. In other words, one molecule that will remain through all your different cycles. So after that, you do the imaging. Like I mentioned, we use the Leica SP8, which has a white laser that spans for 470 to 670. And the software was the ALSX software. After the imaging, then you go bleaching with the lithium boride, and then you come with your different cycles. So once you have acquired all your images from your different cycles, you go to Ziv's NIH GitHub site that is listed in Andrea's paper, and you do your analysis, your aligning and your analysis using his software. This is just to show you. So the first thing that you do is you prepare your controls. Like we do in flow cytometry, you have to read a fluorescence to teach your machine what is a positive and what is a negative sign for that particular fluorescence. We call this in flow cytometry compensation. So you do a similar thing for when you're using fluorescence analysis in the confocal microscope. But what we did was, instead of using one color, one single color control at a time, we used 2 color controls. And what you do essentially is you take fluorochromes that are pretty far from one another in the spectrum. So you minimize spectral overlap, but you can still use the same sample to tell your microscope what is positive for one or for another marker. So in here, for example, you have the 2 colors. You have the cytokeratin in pink, you have CD20 in yellow, and you can tell now your microscope, what is this color and what is this color, what is staining and what is not staining. So essentially you use that to do your minus background. And you do that essentially with all the fluorochromes that you're going to use, using always controls that are far from one another to avoid the spectral overlap. So this is the first thing that you do. You teach your machine what is a fluorescence and what is not, using your different molecules. One thing that we did, so when you do the staining here, Andrea, in her paper, and we also did use this microwave, that is the Pelco microwave. The Pelco is essentially a machine in which you put your sample, your slice, your tissue in the slide with the antibody on top of it, your little solution of antibody on top of your tissue, and you put it in this microwave. And it sort of works as a correlate of a PCR machine. So it changes temperatures in different cycles, and it goes doing the staining for you. So you can do that for your blocking, and following the blocking, you can do that with your antibody mixture. But we don't have a pelco here in the lab. So one thing that we did is we tried also just an incubation at 30 degrees centigrade. But I tell you, the pelco is viable. It's not that expensive. It's $35,000, and it's worth it. It's really worth it. But we also tried 37 degrees in CO2, 5% CO2 for 1 hour, and we compared the two different stainings. So you see here, for example, the staining for HLA-DR, beautifully stained at 37 degrees, and the staining with the same antibody now in pelco that is stained really well. But I can tell you that the 37 even worked a little bit better than what you can see in the pelco here. Down below, though, we have CD20 and cytokeratin. And as you can see, CD20 stains really well. This is a little defect during the acquisition, so I apologize for that here amidst so many experts in confocal microscopy. But you can tell that you have a good staining with the pelco and the 37 degrees, but the cytokeratin really didn't stain very well at 37, as opposed to the pelco that is stained really well. So, you know, it depends on the antibody that you're using. It depends on the condition that you're using. Some work better with the pelco, but some work pretty well at 37 degrees, 1 hour incubation as well. So after we did that, what we did was we validated a huge number of antibodies. So we validated several antibodies into human, different molecules that identify cell populations, functional characteristics of these populations, as well as structural components. So here we have just an example of different molecules. CD68 for staining tissue macrophages. You can see the cells with the spread around them. What you see in blue here is the staining for the nuclei, which is what we use as a fiducial, because the HIST is not bleachable by the lithium borohydrate, and so on and so forth. So you have many different cell markers that you can use, and we standardized over 50 antibodies for different human markers. Here, one that I liked very much, because this is a cell population that I'm very interested in, which is the CD8 cells that you can see here stained in beautifully in pink, and you can see the staining in green for a functional molecule that is granzyme. So you can see CD8 cells that are positive for granzyme, but you can see cells that do not stain for CD8, but that will stain for granzyme. This one is right here, showing that this is a different cell type. In this case here, it's a CD4 positive cell, so it's pretty cool to be able to see. Then you know, once you assemble your images, you can determine what are the cells, what's the percentage, what is the frequency, what is the intensity, and many different characteristics. So we had a lot of fun establishing many different human antibodies. So this is just to give you an example of a cycling cycle 1 for one of the cervical cancer samples. So you can see here the overlay of the many different parameters and molecules that were stained here. So you see an interesting point that I want to show to you here is that we used a same color or a same spectra for 2 different molecules. In this case here, CD3 with Alexafluor 555 and anti-SMA, which is a marker for vessels, with IF 570. So you see that the spectral overlap is huge. They basically are excited and read in the same channels. But what happens is that the CD3 will be on the T cells and the SMA will be in the vessels. So you can actually use that to maximize the number of channels that you can use in your microscope and minimize the number of channels that you're going to use in a microscope, but maximize the information that you get. So obviously, this is yellow here, is a staining for a T cell that is an inflammatory infiltrate in this region of the tumor, but these ones that you see here are the staining for the SMA, which is in a completely different location and staining a different marker. So this helps maximize the use of your confocal capabilities. So it's sort of like the same line, but in completely different structures that you can differentiate by knowing what they're marking for. Obviously, before we did that, we did the single colors for each one of them to be sure that this would work properly, but this works pretty well. So we've been using it so far. This beautiful staining here, and I believe this is the one that I sent to Cristina because I really like this image, is from a heart of a Chagas patient. And let me tell you, these figures that you see here, some of them were taken directly from the screen of the confocal microscope. So there's no processing of these images at all here. So what you see here in terms of background and everything is what we see when we acquire the images here. So what you see in green here is the staining for desmin, which is the cytoskeleton protein for the cells from the heart. You can see here intense inflammatory infiltrate between the different the fibers of the heart, this inflammatory infiltrate containing CD45 positive cells that are stained here in yellow, but also containing CD8 cells, which are also CD45 positive in this intense inflammatory infiltrate. Here again, we put together the marker for CD8 with the marker for SMA, which is also a marker in vessels that you can identify in vessels. So again, in completely different structures, one is a cell marker, one is a vessel marker, but you can read in the same channel and you see the vessels right here and also the cells completely different in the amidst the inflammatory infiltrate. You can see here the nice staining for CD45 in yellow and cells that are CD45 positive and CD8 positive, but you also see cells that are CD45 positive and CD8 negative. So this would be a different mononuclear cell present in this inflammatory infiltrate. So through the cycle one, with as many different markers, you can analyze many, many different things. And again, the Hirst that is not bleachable is our fiducial marker in this tissue here. Then you go and you do your analysis, and this shows you do your bleaching, and this shows you what happens once you bleach the tissue. You take everything in a channel away. So if you take, for example, CD20, you don't see anything in green here anymore. You keep your fiducial markers, so the nuclei are all stained. And here is to show you that some of the markers, some of the fluorochromes, as I mentioned before, are not bleachable. So you should never put them in your first cycle because you're never going to be able to get rid of them. So for example, Alexa Fluor 594 is not bleachable. You cannot use it for your first cycle unless you're not going to use that channel again, right? So it's very important to do this bleaching properly. Here, I show you now a staining for cycle 2 in the very same sample of the cervical cancer that you saw before. So you have here the cervical cancer. You see the structure, the funny structure here that I mentioned to you earlier. You should choose fields with these funny structures because then when you go read, look at them right here. So you can clearly recognize this structure here and this structure here, even though they're stained with one thing here and with a completely different marker here, but you can identify them. Look at what happens. You see the morphology of the tissue, and that helps you choose the area where you're going to acquire your second cycle. So you come with a number of different markers in your second cycle, and now you can have, for example, a total of 11 markers. And as you said, as you saw, you can reach 28 to 37 markers easily in these different tissues. Here is the Chagas heart, in the second cycle for the Chagas heart. So you have the first cycle here, the funny structure here that you can again recognize here, you see, So it's the same funky structure to help you guide where you're going to read. Now you don't see anymore of the green, which is the desmin that you had in the first cycle. But look here in this corner, you see a little bit leftover desmin here, right? So we learned, this is, I showed this image to you, even though this is not the one that is our final, but just to show you what happens. So sometimes you have to use light. So depending, we started using light at all the time to be sure that we would have complete bleaching, because some of the fluorochromes, I think because of the intensity of the staining, as you can see, it's really strongly stained here. You cannot bleach it very easily. So you have to be sure to use the lithium bromide together with a lamp, because then you can bleach it. Okay, so the conclusion here is that, yes, we were able to successfully standardize the use of manual IBEX for human FFPE samples from pathology labs, which made us really happy. We used the pelco, as I mentioned to you, the microwave, but we also used 37-degree incubations that allowed for very good results. We tested a variety of tissues. I showed you here the cervical cancer, the Chagas, but we also had different samples from Leishmania patients, cutaneous, mucosal, disseminated samples. We also had patients, lesions from patients that responded to chemotherapy and did not respond to chemotherapy to see what happens at the site of the lesions. We validated actually over 50 antibodies in this methodology while we were there. We used the SP8 Leica that they had available there, but we also used more simple machines with less ability to read so many different fluorochromes, and they work really well. As I mentioned earlier to you, what will happen is that you're just going to have to add more and more cycles, and this is obviously highly applicable for many different uses. I put down here for you, but this is also in a better, more clearly presented in Andrea's papers, which are the extensions, the programs, the free programs, which is the Imaris reader and the extensions and algorithms that Ziv developed that you can use for analyzing these samples. So we are now in the process of analyzing this. As you can imagine, this generates a huge amount of data. So it will be a while for us to actually have all the cells and the frequencies and the intensities and all analyzed, but we are in the process of doing that. And hopefully, perhaps in the next year presentation, I can show you some of the data that we've got from this. The bottom line here is that, yes, you can use this. You can use just one slice and do many different cycles using fluorescence, and that will allow for you to stain as many different markers as you have antibodies that can be employed, or even different molecules that can be employed here. So with that, I will finish my presentation by thanking Carol, who has been working very hard and has been very instrumental in the analysis using this methodology, Professor Geraldo Brasileiro from the medical school for the partnership with the Chagas heart samples, Ken Golub from Hospital Israelita, Ken is right here, was there working hard with me when we were there in January. And of course, our hosts at the NIH. This is Alan Scher, who many of you know, who used to be the head of the parasitic immunoparasitology branch at the NIH. This is our dear friend and collaborator, David Sachs, Ken and myself, and this is Dr. Giorgio Trinchieri from the NCI, who has also been very instrumental in establishing this methodology and bringing this methodology to Brazil. And obviously, without the help of the funding agencies, the NIH itself, the National Institutes for Science and Technology in Tropical Diseases, and especially in this case, CNPq, who granted me a Estágio Senior Fellowship to go to the NIH and learn it. This was really excellent, and I'm very excited to establish these methodologies here in Brazil. So thank you very much for your attention. Thank you again, Chris and Greg, for the invite to be here today. And I'll be glad to either talk today, or I will be also available in my lab once I'm back in Brazil to discuss this further with you. Thank you very much.
I I am the one who thank you, Val, for the great presentation. High applicability, I think, is the word here, right, Val? I think that many in the audience are thinking about how can I have access to such a powerful technique to apply in their research. And a great thing that from your one of the many great things that you did in your talk was to point out troubleshooting, right? What can go wrong, what can go right, and what can go wrong with this technique. And one thing that people must be thinking is that are you planning or do you have the money already to establish this in Brazil? Is anybody doing this in Brazil?
Oh, this is the wonderful. So this is the wonderful thing of this methodology. So we can read this. We can, as you know, we do have access to confocal microscopes. So we can use the confocal microscopes we have, and you can do it. You can do it using your antibodies, your incubator. You just have to buy the commercial solutions that are free, that are, you can buy them easily. So yes, we are establishing this here. I still want to get the Pelco machine, which, like I said, is not that expensive. It's $35,000 only, and it's truly worth it because it's really fast. You can do one cycle in one morning. You can do one cycle in the morning and then you read the next morning. So this is another catch that I learned the hard way while I was at the NIH. At first I thought I'm gonna stain and then I'm gonna read right away because then I'm gonna have fresh fluorescence. I'll stain in the morning, read in the afternoon. But remember, you have to invert, you put your mounting media, which is not a hard media. It's a media that doesn't get hard because you have to remove it later. You have to remove your cover slip for your second cycle. So it's like a loose mounting media. And when you invert it, if you don't let it sit for at least 5 hours, it's going to give bubbles. So your images are awful. So the first images that I got were really bad. Then I learned that if you leave it from one day to another, just sitting there in the dark chamber and you acquire next morning, when you invert it, it's just beautiful. So yes, we are doing it here. You can do it here. It's feasible and it just works beautifully.
Great news. Do you still have time for some questions, Val?
Oh yes, of course.
So there is a question here. Let me see if I can read. From Valeria. Val, do you lose antigen presence exposure when you bleach? Is there an optimal sequence of staining for different components of cells or ECM?
Okay, this is a good question. It's a very good question. So we actually tested the antigen stability after the bleaching. So we did staining. We bleached and we restained with a different marker, and you can see that you don't lose from most of the antigens that we tested. For all the antigens that we tested, I didn't test all of the 50 different antibodies, but we tested a bunch, about 30 different markers. Andrea tested a bunch, and you do not lose your antigen recognition after your bleaching. Remember that to test that, you have to use antibodies with different specificities because the bleaching does not remove the antibody, it just bleaches the fluorochrome, right? So you can test that, but you need to use different specificities. But no, you don't lose the stability. Regarding the optimal sequence, one thing that you have to be really aware of is that sometimes your antibody is not directly labeled. If your antibodies are all directly labeled, you're fine. You can do that. Unless you're using, for example, CD3 and TCR, structures that are very close that can interfere with the expression or with the recognition of one another, then you have to be careful with the reagent that you choose. But regarding the sequence, what you have to be aware is that if your antibody is not directly labeled and you have to use a secondary, you have to be sure that if you're going to use an anti-IgG1 for a particular marker in your staining, you don't have any other IgG1 in your panel, right? So you have to design the usage of your secondary antibodies according to all of your isotypes that you have in your panel. That's the most important thing for you to be careful with. All the rest, if you have directly labeled antibodies, just be aware of, of something that could impair the recognition of the other molecule. But typically, that's not something that happens very easily, at least with immune cells.
Okay, Chris, can I just make a comment?
Yes, please, Greg.
Val,
You have the word.
Val, so just so you'll know, we have a Pelco in the center of microscopy.
Yes.
So,
That's
so that
fantastic.
should help. I think it's an older one. I have to check. I'm not sure about the programming on it, but Patricia Martinelli she can tell you more about it. Masara can tell you
Well,
more
that's
about
the
it.
best news of this year, Greg. I'll get in touch with you with the specs that we use, the one that we use there. Oh, I'm so glad to hear that.
Okay, just be
Because
sure
that really makes a big difference. That's really a wonderful thing to have.
Okay, just be sure you bring me a bottle of French wine when you come.
Absolutely. I'll bring you some champagne.
Lots of bottles, right?
Yeah, let me just make one other comment to you that I tried a lot of antigen recovery years ago with the Down syndrome tissues and things I collected from pathology. And I always had a really big problem with repeatability. If I took the same sample or sections, adjacent sections, and then the next day, I had a huge problem with repeatability, reproducibility.
Right.
And so it's kind of, I don't know if you've done those tests of like do one day and then the next do another same material, but do it again a second time, whether it's reproducible.
Right, Greg. So yes, we did that. So this is one of the things that Giorgio also mentioned, that they had problem in the past with the cancer samples. So we did that. We tested the AR6, which is the one we used most. And we also test another buffer, which was AR8. So depending on the molecule that you're trying to recover, you have to tweak a little bit whether you use 6 or 8. But we did repeat several times actually from the same tissue with the different slices from the same tissue to try to recover. And it's really, really excellent reproducibility. But I tell you, the reason why I wanted to do that is because this is the part that is most primitive of the entire process. Basically, you put your slide inside of a Falcon tube with the buffer diluted in it, and you boil it at 90 degrees inside of a beaker. And you have to be there looking at the temperature to be sure that it's going to be at 90 degrees. But if it raises a little bit, you put a little bit of water. And, it's this thing that is like really manual and really primitive. But it seems to work really well. So with the molecules that we tested, which were, over 50, we had no issues at all. So it worked really well.
You didn't have any problem with autofluorescence in the tissue, or was that bleached out?
No, that's the other thing. For some markers, so for example, one of the ones that gave us a bit of a trouble was CD68 and CD206, which are two macrophage markers. I don't know if it has to do with the type of cell or if it has to do with the actual antibody. So those you can clean them beautifully with the program that Ziv did. But even when you're looking at the screen as you're acquiring the sample, you can clearly see that you have a good staining, but you do have some background, so you can clean that. But most of the images that I showed you here today were all prior to any processing at all.
Very good. I want to talk to you. I'm not going to take time now, but when you get back, give me a call, because I think there's another method you should think about using rather than antigen recovery, but that also means the way that you prepare your tissue to begin with.
Right.
So,
yes.
But not now. It's like we better go to the next speaker. So,
I'll look for you for some. Perhaps I'll pay you a visit, Greg, at the center, so you show me your Pelco and we talk about it.
Okay, great.
All right.
Thank you so much, Val. I think we could stay the whole morning talking to you here, but unfortunately we are running a little bit late.
My pleasure.
So I wish you a safe return home and a great meeting there in Geneva, where you're heading to. Have fun as well. And thanks again for being with us.
Thank you very much for the invite. It's really a pleasure to be here, and congratulations on the excellent meeting. And I'll see you guys very soon. Okay. And if anyone wants to talk about it more and all that, it's valdutra@icb.ufmg.br. It will be a pleasure to talk with you.
Thanks a lot, Val.
Bye, everyone. Nice to see you all.
So, Monica,